Hello everyone. Thanks for clicking on this video again, and it's another one. The title of this video will be Quantum Mechanics Might Help Solve AD. I know that's quite a stretch there, but I will explain more as I go into the video a little bit. Well, thanks for clicking on it again. I hope that y'all like and subscribe on this video and comment. Let me know if there's anything that I can do to make my videos a little bit more digestible. Try to make them as simple as possible, but if there's a way that I can kind of make them even more simpler, let me know, please. I'd love to get some feedback on that. All right, let's just jump right into it. So, why do proteins in diseases like Alzheimer's, Parkinson's, and prion diseases suddenly become toxic and start building up and start destroying the brain? Sorry. So, to just jump on it, the paper that we're going to be talking about today is going to be explaining why this happens, and it utilizes basically advanced computer simulations to analyze proteins that are misfolded, and it looks at them from a subatomical perspective. So it looks at the electrons and the proton version of these type of proteins. So what is the name of the paper of this week? So this week's paper is titled From Molecular Dynamics to Quantum Mechanics of Misfolded Proteins and Amyloid-like Macroaggregates Applied to Neurodegenerative Diseases. This paper was published in 2022, and it is authored by J-Neuro Gillet. Alrighty. So imagine what if one of the biggest mysteries in Alzheimer's disease starts with a tiny mistake? Not a mistake you can see with a microscope, not even a mistake at the level of atoms, but a mistake involving the behavior of electrons themselves. What are proteins? So imagine you're folding a piece of origami like what is put up here. This is a piece of origami. It's supposed to resemble a cake, I believe. But imagine resembling. You see how elegant, how true to its form it's doing. It's essentially telling you exactly what it's going to be doing, which is basically representing a piece of cake, right? Or a subsection of that cake. That's what it's doing. It's showing you its function. Proteins work in a similar way. Your body constantly builds proteins, and each one must fold into a precise three-dimensional shape. When proteins fold correctly, they perform essential jobs. This is carrying signals between cells, building tissues, supporting brain function, helping neurons communicate. For years, everything works exactly as planned, but sometimes something goes wrong. So what is that? Sometimes proteins misfold. At first glance, this might not seem like a big problem. After all, it's just one protein, but here's where things become dangerous. Misfolded proteins often become unusually sticky. Imagine dropping a piece of gum into a box of Lego bricks. Soon other pieces begin sticking to it, right? Then even more pieces attach. Before long, you've got a giant tangled mess. This is very similar to what happens inside the brain. So when it starts misfolding, when proteins start misfolding, it does not perform its regular tasks. Cell dysfunctions, brain health deteriorates. In the paper, it describes something known as the zombie effect, which is very, very interesting when you look at it from the perspective of this type of proteins called prion proteins, which are usually characterized by prion diseases. A common one is mad cow disease that happened, I think, early 90s, where people just ate wrong cow meat and they developed some neurodegenerative diseases, and eventually most of them ended up dying, unfortunately. So scientists discovered that a misfolded protein can actually influence healthy proteins, which is what happened with those prion proteins, to adapt the same abnormal shape. Think of it like a zombie movie. One infected individual bites another, and that person becomes infected too, and the cycle continues. So a single misfolded protein can trigger a chain reaction that spreads through a population of healthy proteins. This creates more and more toxic proteins over time. So as these misfolded proteins accumulate, they begin forming clusters. Scientists call the smallest clusters oligomers. So I want to explain exactly how this pathway happens, especially specifically to Alzheimer's disease, and how amyloid beta becomes the destructive protein that it is. So in normal circumstances, on the left part of this illustration here, we have a cleavage of amyloid precursor protein by alpha secretase, which is a normal pathway that produces soluble amyloid precursor protein beta, which is a harmless peptide. It also there's another protein process that is known as the gamma secretase that comes to cleave the other remaining membrane-bound protein of the amyloid precursor, and that's CD3, and that is basically cleaved by gamma, and it forms AICD, a very neuroprotective protein that goes onto the nucleus and causes neuroprotective pathways to be activated. But in the amyloid-degenerative pathway, instead of alpha secretase, we have beta secretase. This cuts it, and it produces soluble amyloid precursor protein beta, which is then cleaved by gamma secretase, causing the production of AICD as well as amyloid beta, A40, A42. So these amyloid beta proteins are very sticky. When you look at beta sheets, they tend to be very sticky because they have a lot of hydrogen bonds sticking out, while the hydrophobic ends, they kind of fold into the protein itself, because the neural environment is very hydrophilic. So the hydrophilic hydrogen bonds are going to stick out in most of these beta sheets. So when they stick out, and then they see other amyloid beta proteins that are just kind of in the lumen, they're going to cause them to also stick to them, and eventually lead to the formation of the clusters or the oligomer structures that are being talked about here. So as additional proteins join, these clusters grow into long fibers known as amyloid fibrils. Over time, these fibrils can combine into massive aggregates, and these protein deposits are commonly found in several neurodegenerative diseases. For Alzheimer's disease, we know them as amyloid beta and tau proteins. Parkinson's, it's alpha-synuclein. Prion disease, it's usually prion protein itself. Different diseases, different proteins, but surprisingly similar processes. Misfolding, aggregation, and there we go, brain damage. So you have a healthy protein becoming a misfolded protein, and then more misfolded proteins arise from that. So for decades, researchers have used a technique called molecular dynamics. Think of molecular dynamics as a supercomputer generated movie. So it basically just looks at proteins over time, and the simulations have been incredibly valuable. So they help researchers understand how proteins fold, unfold, and interact with one another. But according to the authors of this paper, there may be an even deeper level to the story. As I've already explained, when proteins misfold, the structure changes, they develop large regions rich in beta sheets. These beta sheets act like molecular Velcro. The proteins begin sticking together. Aggregates grow into amyloid fibers, so I kind of got ahead of myself there. But essentially, that's exactly what they found in the paper. So quantum mechanics studies the behavior of electrons, the tiny particles that surround atomic nuclei, the subatomic particles. So instead of asking where are the atoms moving, like what molecular dynamics would have asked, which is what is kind of the standard right now, quantum mechanics asks how are the electrons interacting? Why does this matter? Because every force that causes molecules to attract or repel one another originates from interactions between electrons. The authors argue that if we want to fully understand why amyloid proteins stick together so strongly, we need to understand these quantum level interactions. In other words, the root causes of protein aggregation may begin at a scale far smaller than we normally considered. The paper highlights structures called beta sheets, which is what we're talking about here. So imagine two strips of Velcro. That's exactly what these beta sheets are. They're just sticking to each other like two sheets of Velcro. And if you have multiple Velcro, it's going to be clumped up. And if this is inside the brain, you can see how it can stop a lot of the processes that are normal inside the brain, right? So it's going to disrupt ion channels. It's going to cause a lot of oxidative stress based on microglia trying to clear them out, and them attacking things that they're not supposed to be attacking, or attaching to things that they're not supposed to be attaching, for leading to further stress inside the entire brain environment. So the researchers suggest that quantum level forces may help strengthen these interactions, making amyloid fibers more stable and harder for the body to remove, which is why we need to find more therapies that are kind of linked towards targeting these specific subatomic structures of amyloid beta. And so why does this matter? This matters obviously because we want to be able to develop medicines that are actually going to target these structures, right? We need them to target these structures more precisely, sparing the non-misfolded and otherwise healthy brain environment. And it's important to also understand that subatomic interactions can help reduce the structural formations before they start, and keep the immune systems in the brain trained to recognize them and delete them before they cause harm, right? So once we understand this interaction, The scientists can determine exactly how toxic aggregates form. They may be able to stop the process before severe brain damage occurs. The knowledge could help researchers design better drugs, so more good antibodies, immunotherapies, training the microglia to kind of to target these structures before they form and before they oligomerize to become fibrils. And therefore, so the main takeaway is that the authors argue that understanding neurodegenerative diseases requires looking at multiple levels of biology. The toxic protein aggregates associated with Alzheimer's and other neurodegenerative diseases might ultimately be driven by physical forces operating at the smallest scale of nature. Understanding these forces, scientists may uncover entirely new ways to fight these devastating disorders. And that is the main takeaway that I want to leave for this video. Having precise treatments is the best way forward for this disease, and reducing the chances of harming the neuronal environment, because it's such a sensitive area, is the most important thing. So if this disease can be treated at such a complex level, it might help actually come up with treatments that are very much targeted towards the cause instead of the overall symptom alleviation aspect. And even, who knows, once we're able to solve Alzheimer's, perhaps we can solve a lot of other brain diseases and even improve brain health across time. All right, thanks for watching, and we'll see you in the next video. Again, please like and subscribe and comment. Let me know if there's anything that I could change. All right, take care. Bye.